Today is the 13th October 2021, and it's my pleasure to have Jason Shepard from University of Utah, USA. So Jason is an associate professor in the Department of Neurobiology and holds the John M. Huntsman Presidential Chair at the University of Utah. He obtained his Bachelor of Science with honors at the University of Otago and his PhD at the Johns Hopkins School of Medicine and postdoc training at the MIT. So he's a recipient of multiple awards, including the Peter and Patricia Gruber International Research Award in Neuroscience, the International Society for Neurochemistry Young Investigator Award, the Chan Zuckerberg Initiative Ben Barres Early Career Acceleration Award, the NIH Director Transformative Research Award, the Research to Prevent Blindness Tain Innovation Award, and Eastern National Academy of Science Kavli Fellow. So his lecture today is going to be about virus-like intercellular communication, a new EV paradigm. So this is something a topic that's quite hot right now, and I'm really looking forward for his talk. So before we started, I'd like to thank Horiba Scientific for supporting this session.
All right, thanks very much. Caroline said I should give a little bit of an introduction to my path. And so I actually grew up in South Africa and then moved to New Zealand when I was 14. So my family's originally from the Southern Hemisphere. And then I went to high school and undergrad in New Zealand, and originally was trying to decide between medicine and neuroscience, or medicine and just doing research, and did a bachelor's, ended up just deciding to do a bachelor's in neuroscience. And when I was looking to do research, we had an exchange agreement with the University of California. And so I spent my honors thesis, my honors year at UC Irvine. And that's how I was able to get access to applying for grad school in the States, and ended up going to Johns Hopkins for a PhD, and then stayed basically in the States since then, and was a postdoc at MIT for 5 years. And then after that moved to the University of Utah, where I've been just over eight and a half years, where I started my own lab. So that's my quick path. And as I'll sort of describe, we're fairly new to this field in terms of the extracellular vesicle field. And it's not been a focus of my lab. The questions that sort of are important for originally important to the lab was really, and still are, is how memory works at the molecular level. So how do you store information in the brain? But I'll describe a little bit of how we got to where we are right now in terms of the EV field. And I'm not sure if you guys have questions or live questions during the talk, but happy to take questions as well, or we'll just wait until the end. Okay, so of course, all the work I'm going to tell you about is not done by me. It's done by these guys in the lab, and I'll point out who did the work as we go along. So as I mentioned, really the biggest question in the lab that sort of drives the overall big picture is, how do you store information in cells in the brain for up to a lifetime, despite the fact that most proteins turn over within minutes to hours? And so neuroscience have been searching for the long-term substrate of memory for many years. And we, of course, can take multiple approaches to understanding this, everything from looking at circuits. So these are neurons in an intact brain as we're imaging into the brain. And this is in a mouse brain. And these circuits are made up of cells that are connected with each other through synapses. And we know that these synapses are structures that can change their function and structure when you learn. And there's a specific set of genes and proteins that can regulate these synaptic connections, and ultimately sculpt these circuits that are specific to the memories that are encoded during training and learning. And of course, there's a temporal order to all of this. And when you learn, the first thing that happens is, of course, electrical activity. You get action potential firing, synaptic transmission, and that results in various ions that are conducted through channels. And the electrical conductance, of course, is what propagates the signal. But at the same time, you also get these second messenger cascades that can also occur at certain synapses that are driven by calcium. And there are some classic then kinase second messenger cascades in neurons. But ultimately, for memory and learning, we think then that these signaling cascades induce a program that's required for consolidation of memory. And so the big question then is, what are those genes that are induced, and what are the proteins that are important for these kinds of processes? So on the other hand, so you've got the temporal dynamics that need to be sort of understood, but then you also have the spatial scale. So one neuron here, for example, in the pyramidal type of neuron has about 1,000 or so synapses. So there's each individual synapse needs its own repertoire of proteins that can regulate these synapses. And so how do you do that? One way is that you can have transcription and gene expression, but the RNA actually doesn't get transcribed immediately. It gets trafficked out into these dendrites, and then it can actually replenish and change the composition of proteins at synapses in a very specific way. So these are just some of the unique aspects of memory consolidation. We also know that if you have mutations in genes that can that are in the signaling pathways that lead into these processes, you get intellectual disability and cognitive dysfunction and autism spectrum disorder. So all these red boxes are known human mutations. But what's key is that they all sort of cascade into only a few proteins. And so we've decided to sort of look at what are these main effector proteins, what are the proteins that are at the end stages of these cascades, and what is their function. So just to summarize them quickly, we think that when you learn, you turn on a key program of genes. Some of these genes have RNAs that are transcribed but not necessarily translated. They get transported out into synapses and active dendrites, and then when they're translated, they can actually regulate the function and structure of synapses and really change the composition of what those synapses are made of. So as I mentioned, you could take many different approaches to this. We've taken a very narrow approach to studying this, and I've looked at this one particular gene for most of my career called ARC, which stands for Activity Regulated Cytoskeletal Protein. And it was cloned at roughly at the same time into two different labs. So Paul Worley at Johns Hopkins and Dietmar Kuhl, who called it ARCH 3.1, were looking for genes that could be induced by neuronal activity. And this is actually a rat brain where Paul and Oz Stewart did in situ hybridization experiments to look for RNA, ARC RNA. And you can see here on the left, this is a rat that was sacrificed in its home cage, and this was a rat that was allowed to just explore a novel environment for an hour. And you can see here, of course, that there's massive induction of transcription, and a lot of the mRNA that is induced is in the cortex of the rat and in the hippocampus of the rat. And these are structures that we know are important for memory. This C-shaped structure is the hippocampus again, and in humans, all the way down to all mammals have this structure. And if you take out the structure, you cannot make new memories. The other interesting thing about this in situ is that you can see here in the blow up of the hippocampus, this fuzzy staining here is, again, those dendrites. So the cell body layer is these dark dots, and then you can see this mRNA in the dendrites. And this was one of the first examples of a robust expression of an RNA in these processes in the dendrite. And so then Oz Stewart and Paul went on to show that the induction of this RNA is very dynamic. You can induce it in the cell body layer, but if you stimulate with an electrode just a certain set of synapses here. You can see this very well demarcated line where those synapses were active, and the mRNA is trafficking just to those active synapses. And so that, of course, implies that you can get this rapid translation of protein. This experiment from Kim Huber's lab, where they took those hippocampal structures, made brain slices, and actually severed the dendrites away from the cell body, and then quickly put an agonist, this drug here, DHPG, which is a metabotropic glutamate receptor agonist, induces translation. And you can see here in this image that there's a robust increase of ARC protein, even in the dendrites that were severed from the cell body. So definitely saying that you can get translation of the RNA in these dendrites. So again, in neuroscience, because the hippocampus is so important for learning and memory, it's been used for a long time to study these synaptic changes. And so the best model that we have for memory really comes from studies where they've looked at synaptic plasticity in this pathway in the hippocampus, particularly the CA3 to CA1. So these are synapses that leave from the CA3 and then synapse onto the cells in CA1. And the details are not important, but this is to say that you get potentiation or depression, so basically weakening or strengthening of these synapses in response to particular kinds of neuronal activity. And so one of the functions of ARC that we know of, and this basically summarizes my PhD in one slide, that we found this ARC protein can actually interact with these endocytosis proteins, endophyllin and dynamin, to recruit the APA-type glutamate receptors. This is the GLU-A1 or GLU-R1-2 receptors. And so when you have lots of ARC in the synapse, you get less receptors at the synapse, and the receptors get recruited out of the synapse. And so you get weaker synapses, or LTD. On the flip side, so now we know all this, this regulation of this gene, it's turned on during memory, it seems to regulate synaptic strength. But if you take it out of mice, basically they don't learn anything. Their short-term memory is intact. So if they're just after training, they seem to have learned something. But if you come back a day later, there's basically very little recall, and all the sort of kinds of memory tasks that we can subject a mouse to without asking the mouse what it actually can remember. So this is a spatial memory, fear conditioning, where you pair shock with a context, or conditioned taste aversion, where the animal learns that a certain taste is aversive, novel object recognition, so whether it's known that it has seen something in the environment that it's been in. So that's kind of a big severe defect in memory, which is good if you're trying to understand memory. But there's just a little bit more background here. So it's a single copy gene, but I'll explain a little bit about the evolution of it. It's a classic immediate early gene, which just means that its synthesis, its transcription does not require new protein synthesis. And we've been known for a long time that it's highly dynamic, that when animals are learning in a particular kind of behavior, you can get this gene turned on. And then you also get big other immediate early genes like CFOS in the brain that are transcription factors. And what was sort of puzzling is that this gene is not a transcription factor. It's predominantly expressed in the brain, and except for a few other cell types. And then within the brain, it's only in a subset of these kinds of neurons that are excitatory, and it's not expressed in glial cells. So it's really quite specifically expressed in neurons and a certain kind of neuron. So that sort of summarized what we knew about this gene as I started my lab. And there was a set of experiments that I wanted to try, which was to try and figure out more about this ARC and glutamate receptor trafficking cycle. But when we started to do some in vitro assays, as I'll get into, we were getting these weird results. And of course, and often in science, it's the weird results that you want to pay attention to. And so this work was originally published in 2018, and it was driven by Alyssa and Cameron in the lab with help from Rachel. But it started off with my first technician, Nate Yoda. And again, what we wanted to do was purify protein. So in this case, purify Arc protein, purify some of these endocytic proteins like dynamin, add some lipids, and then do some in vitro kind of assays to see how or whether Arc could catalyze the activity of these endocytic proteins. So to do this, we used a classic tagging affinity purification protocol. We put a GST tagged Arc into E. coli and then produced a ton of the protein in bacteria. And we were able to then get a pure population of protein after cleaving the tag off the column. And so at this point, we thought, great, we've got this working. And then the last step to clean up the protein is that you run it through a size exclusion column. And that normally cleans up a lot of the bacterial contaminants. And that's when we start to get some weird results in that this is where we expected everything to come out. This is the predicted size of the monomeric arc. But most of the protein was coming out in this fraction, which is the void fraction, which suggests it was so big it wasn't even getting through the column at all. So Nate, at this point, thought, okay, maybe we just didn't know how to purify protein, or we just had the wrong conditions. And so he tried a bunch of different buffers and pH, salt concentrations, and almost nothing really changed this. And so finally he gave up and said, okay, well, I want to learn how to do electron microscopy. And when I just look at the protein that I'm collecting and you know, it's probably a mess because it's probably aggregated or something, but let's take a look. And these are literally the first images we got from that experiment. Of course, when you look at these, you're like, well, what the hell are these soccer ball things? And when we saw this, we were quite surprised, of course, because we thought, well, this is just Arc protein, and what could possibly be happening here? And we showed these images to a few of the biochemists in Utah, and they were like, is your bacteria contaminated with the virus? Because these look like viral capsids. So that one observation led us down this rabbit hole. So we did indeed show that or find that these structures, which are fairly large, around 25 to 50 nanometers in diameter, are made up of Arc protein. And indeed, they do look like viral capsids. And in fact, if you look at EM images of purified GAG protein, which is necessary and sufficient to make the HIV capsid, they're very, very similar to the capsids, these structures that we're seeing with Arc. And while we were doing these experiments, in fact, Paul Worley's group had a paper out in Neurons showing that the structure of a part of Arc is actually just like this capsid domain in HIV. And so at this point, it was all starting to make sense. Now we really were fascinated by what was happening here. So Arc protein seems to have structural homology to a retrovirus, and it seems to be able to actually make retroviral capsids. But it also begs the question of how and why this gene has these structures. And in fact, even back in 2006, a group had predicted using computational approaches that Arc does have homology to these retroviral proteins. Now I'm showing this as well because in the same paper, they predicted that there was a whole bunch of families of genes that also had the same structures potentially. And as I'll get back to this at the end, but basically all the biology I'm describing here for Arc could actually be retained in these other genes as well. And so we collaborated with Cedric Pichot, who's an expert in the evolution of transposable elements and retro elements. And he and his student, John McCormick, did some phylogenetics to sort of try and figure out what was going on here in the evolution of this gene. So the mammalian gene that we're studying is highly conserved between humans and chickens and birds. but there's no equivalent in fish. So the closest sequence that's actually in fish is this active gypsy retrotransposon. We know it's active because it still has these LTR sequences that flank the encoding, the open reading frames. And so the simple explanation or interpretation here is that the ARC gene seems to have evolved from a gypsy style retrotransposon, and this was a transposable element that went germline somewhere in between amphibians and land animals, or fish and land animals. And that's where that gene comes from. So it's a repurposed retrotransposon. But another surprise was that we knew that there was a fly homologue of ARC, and there was another group that actually had shown, Vivian Budnick's group, that had shown that the biology of the fly gene and protein is quite similar to the mammalian gene. So where did the fly gene come from? And this is actually another fascinating observation here, which is that the fly lineage is highly conserved. So there's an ARC or even two ARC genes in each of the lineages that have been looked at. But there are insects, the closest insect relatives, ants, beetles, do not have an ARC gene. And again, the sequence that's closely related are these retrotransposon sequences of the gypsy family. So the interpretation here is that basically this ARC-like gene has evolved at least twice in evolution, probably more. We just haven't sampled many genomes. And strikingly, these are not ancestral to each other, but they do share a lot of the biology, as I'll get into later. And this is really just an, I think, emerging field that these transposable elements can act as sources for new genes through evolution. So being a neuroscientist, of course, I did not know a lot about retroviruses, and so I took a crash course in this with some colleagues here at Utah. And I'm going to give you a quick 101. So in an infected cell, in this case, HIV, the infected cell will make a ton of this GAG protein, and this GAG protein, in a series of different reactions, starts to form a viral capsid. And as it forms the viral capsid, it recruits its own viral RNA, preferentially encapsulating it in the capsid. And as it does that, it actually invaginates out of the cell through a series of lipid reactions. And this is driven by host cell complexes like the escort proteins, which I'm sure many of you are familiar with. And the final particle, the infectious particle, then contains the capsid, the RNA, some transmembrane proteins that are encoded by the virus, like this envelope protein. And of course, there's a membrane around the capsid. So we took a very naive approach to saying, okay, well, if ARC can form these capsids that look like these retroviral capsids, what other aspects of this life cycle can ARC actually do? And so we asked these sort of four simple questions. Can it self-assemble? So we already know that, but can this assembled capsid bind nucleic acid? And can it get out of cells? And then if those two are true, can it actually transmit genetic material, RNA, from cell to cell? And so these are big questions, and these are still ongoing questions in the lab. And I'll sort of tell you about our progress in trying to understand some of these questions. This is taken from a recent review, if anyone's interested in looking at this in the Trends in Neurosciences. So this assembled capsid seems to behave quite well under ice conditions. We've collaborated with John Briggs and his group to do Cryo-EM of these capsids to reconstruct the structure. And as I mentioned before, the fly protein, when you purify it, can also form these capsids that look very similar to the mammalian protein. And for whatever reason, so far we've been successful in being able to reconstruct the capsid in the flies, but not yet in the mammals at atomic resolution. And this was published last year. So this is work from, again, Simon Erlandson and John Briggs's lab. And this is to say that these structures that the capsid are forming look just exactly like a virus. In fact, if you didn't know that this was from an animal protein, John said he was like, this, I would have thought you discovered a new virus. So these capsids are very, very, got, structures that are quite similar to structures that can be seen in viruses, and that's helping us sort of determine some of the biology here. And I won't go into detail of the structure, but you can look it up in this paper. So it's forming these structures. Of course, there's still a lot of questions about how it forms these structures, where in the cell it can actually do this. This has not been as easy as I had thought to sort of do. And so right now we're doing CLEM, so super resolution or light microscopy coupled with electron microscopy. And of course, we want to know a bunch of questions about how the capsid in situ is regulated. So in this purified preparation, that seems to be its natural state, but we think that there's other cofactors, post-translational modification of the protein that can regulate when and where you make these capsids. So of course, then we want to do these capsids encapsulate proteins. And so we took those purified capsids from bacteria and looked for RNA. We thought if they're gonna encapsulate RNA, it should be these mRNA, the highly abundant mRNA that's being made in the bacteria, including ARC, of course. So we can, using RT-PCR, qRT-PCR, we can see robust ARC mRNA expression. And if you add exogenous RNAs, which should degrade RNA, there's no difference in detectability, suggesting that indeed there's encapsulation. Interestingly, if we looked at highly abundant bacterial mRNA, you could see similar phenomenon where that seems to be protected. This is just a positive control to say that if you added exogenous GFP RNA to the same test tube, it gets degraded. And so then we, of course, wanted if ARC protein can do this in vivo. So this is somewhat of a crude experiment, but we took cortex from mice, did immunoprecipitation with an ARC antibody to drag out the protein, and then looked to see if that complex actually has ARC mRNA in it, and it does. So a really robust detection of ARC mRNA in this complex. So that at least says that in vivo ARC protein can be tightly bound to its mRNA. Of course, it doesn't say if this is in a capsid form or if it's protected, but it does say that it can happen in vivo. So some of the other ongoing questions here that we're still trying to figure out is there a preference for a certain kind of RNA? Is ARC mRNA really the main cargo that's being encapsulated? What part of the protein can actually mediate the protein RNA interactions? As in a sort of translational application, we're actually using RNA and gene delivery methods, potentially using these endogenous capsids, and that's something that's sort of ongoing outside my lab. People may have actually seen Fang Zhang publish a paper in Science a few weeks ago on a similar kind of idea. So, as we've talked about, viruses use the host cell machinery to get out and to make their own viral particles. And there's sort of a continuum here between the biology of these viral particles and the extracellular vesicles. And of course, most cells, as you know, produce these extracellular vesicles, and there's a few pathways that are required for their biogenesis, including escort-dependent processes, both through the classic exosome pathway, where you get invagination into the MVB, and then bulk release, or even just budding off of vesicles from the cell surface. In neuroscience, I would say this is an emerging, of course, emerging topic in that it seems to be that extracellular vesicles can be mediating normal signaling between cells, but also abnormal signaling, or act as a mechanism to actually eliminate toxic proteins from cells. Many of the classic neurodegenerative disorders like Alzheimer's and Parkinson's are thought to occur through misfolding and toxic protein formation. And most of these toxic proteins like alpha-synuclein, A-beta, have been implicated or found in extracellular vesicles. And that in itself sort of suggests that this may be a target for therapy. So we, of course, then thought, okay, if ARC is getting out of the cell and it's got a capsid, it should have a membrane around it, just like other viral-like particles, and it should be in the extracellular vesicle fraction. So in this experiment, we took media from primary cultured neurons and then did the classic ultracentrifugation, high-speed ultracentrifugation to fractionate the media. And we did another thing here. So this is the cell lysate. This is ALEX, which of course is a classic escort protein that's a marker for the extracellular vesicle fraction. And we added this drug called bicuculin that will upregulate activity in the neurons and that will induce protein expression, which is what we see here. And what you see is that there's a fairly robust release of ARC protein even under basal conditions that's further enhanced by adding this drug that induces ARC expression. So we think then that ARC is being released and it's probably in some sort of extracellular vesicle fraction. In that same fraction, we can see ARC mRNA, and that RNA is protected against exogenous RNAs, except when you add detergent, and that gets rid of that protection. So we think then that there is a membrane involved. And right now, of course, the big question is what kind of extracellular vesicle, what sort of membrane-bound particle is this ARC-containing vesicle? And so this is one experiment where we've taken that extracellular vesicle fraction and then did silver staining and immunogold labeling. In this field of view, you can see a bunch of different kinds of vesicles, but there's only 3 gold particles. And so we think, of course, just like other cells, there's a very heterogeneous fraction of vesicles. But in the ARC-containing vesicles, it looks like probably you only have 1 capsid. So this is a large gold particle, and there's this high-density staining. The membrane looks like this, we think, a little bigger because it's sort of distorted by the fixing that we've done. But it does suggest that these ARC-containing vesicles have a capsid in them, and there's probably some other cargo as well. So another way to fractionate, of course, media and other sources of extracellular vesicles is by size and density. Here we are using the ISON mini size exclusion columns. Again, we collected the media. The nice thing about the ISON columns is that you can fractionate out the high concentration of junk of the protein, the naked protein, the protein that's just in the media from the more dense or higher buoyancy fractions where we think the vesicles are. And so ARC can be released in these sort of higher fractions. It's not just coming out as junk or naked protein. But we're still trying to figure out protocols to really isolate these vesicles, including isolate them from brain tissue itself. So this is either human brain or a mouse brain. And Radhika in the lab, a newish postdoc, is doing these experiments where she can dice up the tissue and then basically try and dissociate the tissue, extract the extracellular vesicle fractions from that, and then further characterize them. And so we've pretty much got these 2, we think, crude fractions, a large EV fraction and a small EV fraction. And Radhika's in the midst of characterizing some of these vesicles. So this is just EM. So as you'd expect, there's these large membrane structures in the large EV fraction. And then in the small EV fraction, you've got these smaller particles. And we're still trying to see if we can further refine this in terms of making these fractions a bit more homogeneous. But in terms of sort of the classic markers that you would expect to see in these EVs, most of them are present. So CD9, especially these small EV fractions, the CD9, ALEX. We've also looked for CD81, CD63. So we think that we can isolate these fractions, and then we can further clean them up also by running them through the ISON columns. And then we can look at other proteins of interest, of course, like ARC. So we can see ARC out of the in these fractions, and especially quite clearly in these small EV fractions. along with other proteins of interest that we're interested in, like APP and tau, which are implicated in Alzheimer's disease. So some of the questions that we're still trying to address here are, what is the intracellular trafficking pathway that ARC actually gets released through? So does it go through a classic exosome pathway, or is it more of a direct budding mechanism? So we're still trying to test that. Of course, we want to know exactly what's in these EVs. Of course, the challenge here is that we don't have a surface marker, so we can't immune purify the ARC vesicles just yet. So we're sort of doing unbiased approaches like proteomics and RNA sequencing to determine the cargo. And hopefully we'll have some of that done in the next few months. In terms of other questions, of course, we want to know where exactly does this occur, this release in neurons. Does it happen in the cell body or in those synaptic connections, as I mentioned? What controls release? Is there a sort of activity, a neuronal activity that turns it on? And then ultimately, if we can purify these vesicles, can we do structural biology, Cryo-EM, to look at what they actually look like? So finally, given that we've shown that ARC can form these capsids, it contains RNA, and they get out of the cell, can they actually transmit this RNA from cell to cell? So these experiments, we used ARC knockout primary cultured neurons. These are hippocampal cortical neurons, and used them as our recipient cells. We had wild type or knockout cells as our donors, and then we would extract the extracellular vesicle fraction from those neurons. In this case, it was just a crude sucrose pellet ultracentrifugation pathway, because that protocol, because that allowed us to get the most protein. So when we incubate those extracellular vesicle fractions, the wild type EVs do allow transfer or uptake of ARC protein even within an hour. And when we look at the ARC mRNA, of course, there's no knockout, there's no mRNA knockouts. This is what the wild type ARC mRNA looks like. It's quite distributed across the dendrites. And we finally also show that if you incubate extracellular vesicles in these knockout neurons, you get uptake of the protein, and then finally you get transfer of the RNA into the cell, and this is quite robust. So what's interesting here is that we get fairly low levels at even 1 hour, and that's partly, you think, because as the protein gets taken up, it still has to actually disassemble and escape the endosome for the probe to access. And so this is I think something that we're sort of exploring right now, is sort of the dynamics of that path, how exactly the capsid disassembles. And then finally, if there is transfer of the mRNA, it should be able to be translated. And so we use that trick that I mentioned earlier, where we add this drug DHPG that we know induces translation of endogenous Arc. So in the knockout neurons that receive wild type extracellular vesicles and the drug, we see a further increase in ARC protein levels in these dendrites that's blocked by a protein synthesis inhibitor, cyclohexamide. So suggesting that the transferred RNA can be translated and can escape the endosome. So this is just to summarize what, some of the questions and where we're going with this. There's two sides of what we're sort of really trying to figure out in terms of the cell biology. That's, of course, how does the ARC protein capsid get made and assemble, and what are the RNAs that are encapsulated? And then what are the release mechanisms? How does it get out of the cell? What are the proteins that are on the outside? Are there specific transmembrane proteins? And then on the flip side, on the uptake side, of course, we want to know what is the specificity of uptake? Is there a receptor involved? We know this part of the process, this uptake, is endocytosis dependent. If you block clathrin-mediated endocytosis, you don't get any uptake. And then finally, of course, we want to know, how does the capsid itself disassemble and allow the cargo to actually get out into the cell? And then what is that cargo doing in the cell? So what is the functional relevance of this pathway? This is a little bit more neuroscience, and we're still sort of on the hunt to try and figure out what this pathway is actually doing. There's sort of two main models. One is that it's this mRNA, so ARC is transferring its own mRNA to cells, and then when it's translated along with the broken down capsids, that ARC protein can affect the strength of these synapses that take up the capsid. And that could be important for the connectivity that's involved in specifying what memories are encoded. Alternatively, but not exclusive, not mutually exclusive, you could also have small non-coding RNAs like microRNAs or even tRNAs that are being transferred by these vesicles and capsids that could change, gene expression, change protein expression in recipient cells. So that's still to be determined. So just to then recap, some of the big picture questions we're still trying to address is, what are the cargo? Where does this happen? What are the intracellular pathways that feed into this? What are the uptake mechanisms? And then again, from a functional point of view, do we even require the cell-to-cell signaling in memory consolidation? And finally, sort of a more translational aspect of what we're doing potentially is to see if we can use these endogenous capsids for gene delivery. And so I know I threw a lot of information at you, and so I'm just going to end here with a brief animation that that hopefully will be loud enough for you guys to hear. But if not, you can at least hear see the sort of ideas that what we think is going on.
Activity-regulated cytoskeleton-associated protein, or ARC, is a key regulator of long-term synaptic plasticity that is essential for memory consolidation. In the mammalian brain, the transcription of ARC is turned on when we learn new things. Within the hippocampus, the part of the brain essential for making new memories, excitatory pyramidal neurons in the CA1 region have an important role in memory formation. In CA1 neurons, some ARC mRNA is transported to dendrites, where local translation into protein can occur. Newly translated ARC monomers assemble into a virus-like capsid that packages ARC mRNA and other small non-coding RNAs. Alternatively, the capsid may form a vesicle that invaginates into a multivesicular body, which fuses with the plasma membrane and releases vesicles containing the ARC capsids. The vesicles then travel to a neighboring neuron. The uptake of the vesicles is endocytosis dependent. The ARC mRNA is then translated into ARC protein. ARC monomers disrupt the connection between the scaffolding proteins Stargazin and PSD95, which allow the AMPA-type glutamate receptors to move out of the synapse. ARC recruits the endocytosis protein AP2, which binds to the AMPA receptors and induces internalization. Reduction of AMPA receptors at the synapse decreases synaptic strength, which may allow a specific set of neurons to be incorporated into the circuit that mediates a particular memory.
All right, so with that, I'll just end again with thanking the people who did the work in the lab. These are the folks in my lab and our excellent collaborators at Utah and outside of Utah. And of course, our funding mechanisms that allow us to do the work. And so with that, I'll take any questions, and thanks for attending. I know it's a different time zone and all that.
Yeah, thank you so much, Jason. Wonderful presentations and really exciting data that you presented today. So I'd like to open the Q&A session. So if you have any questions, please go ahead and jump in. Just turn on your video and your microphone.
Am I on? Hey.
Hi, Michael.
Hi, Jason. That's just fascinating. One of in the early work that was done, say proteomically, some things people haven't talked about, but it certainly was noticeable to me, was the number of proteins that would form oligomers under normal conditions and so-called normal conditions in the cells that seem to be showing up in the vesicles. And I just kind of wonder if this is more of a standard topic in the field than we think about. And in particular, when you're pointing out the capsid proteins here, it seems like this sort of self-organization may be a much more intrinsic form of communication, cell-cell communication, than we've sort of given it credit. And I'm not saying that everybody's loaded with GAG proteins and all the cells are spitting those out, but it's certainly, this is really intriguing in that. And in particular, I mean, I've got a billion questions, but I won't take up all day, but just one right off the ARC protein associated with its own mRNA, do we know how or when this occurs in the cell? I'm envisioning this occurring, say, at translation, when it's like, okay, and it decides to grab onto its own message, and then the ribosomes fall off, and now you've got it put together, or do they have to dissociate first and come back together?
Yeah, no, exactly. Good questions. And we do think that this is sort of maybe the tip of the iceberg in this kind of signaling. I should put this in, but it was sort of a couple of weeks ago now. Fong Zhang published a paper in Science showing that many of these GAG-like genes in mammals can get released from cells. And the purpose of that paper was sort of more translational and looking at how you could potentially use them for, transfer of RNA. But the sort of main findings were, and we have unpublished data that's sort of similar, suggests that there's other genes that are also capable of making these capsid-like structures. And so then there's going to be, unique cargo, unique cell type specificity. And so we think this is a sort of new paradigm of signaling, and that it may not just be, this unique ARC pathway. But yeah, so back to the model of how the capsids assemble. Right now, we do have this sort of idea, as you sort of mentioned, that it's this co-translation, and we know that the ARC mRNA, a lot of the ARC mRNA is being trafficked out into these dendrites, and then it's poised for translation in these granules, these large RNA granules. And so it's not immediately translated. So you can get this sort of bolus of translation with the right signal. And then, as you say, you can basically grab onto the RNA as it gets formed, as the capsid gets formed. But that step there where, how does the protein tell that it needs to be forming a oligomer? We're not sure exactly, how that works. And in HIV GAG, there's sort of seeding events that happen at the membrane. So it could be that there's a similar kind of thing where you have a part of ARC protein that gets seeded at a particular membrane, and that helps form the capsid. And so we're looking at, for example, palmetylation. There's some cysteines that can allow that to happen, stuff like that.
Very cool.
Next question, please go ahead. Maybe Philip.
Jason, great talk.
Hi there.
So I have a question, because you talked a little bit earlier about other places where ARC is being expressed, and I wondered if you could elaborate on any other known phenotypes that you might see in maybe like an ARC knockout, or just any other known functions that you have about ARC, because, for example, it's expressed in dendritic cells, which I thought was also pretty interesting. So I'm kind of curious what the landscape of ARC functionality is outside of the brain.
Yeah, so there's been one published paper so far looking at immune defects. And so there are there seems to be immune defects in the ARC knockouts, the sort of germline knockouts. In that paper, they were just sort of looking at this EAE model of multiple sclerosis, where you can get this T cell invasion into the brain, and there's sort of if anything, the ARC knockouts had a protected, like they had less severe immune activation, but I'd say there's a lot there to figure out. We know that it's expressed in these dendritic cells, but then of course the question is, can you get release of ARC from those cells? We just started a collaboration with Ryan O'Connell here at Utah to try and actually look at that. He's a immunologist or pathologist that's looked at the role of extracellular vesicles in the immune system. So they could be a source of these vesicles outside of the brain as well, for sure. But in terms of how the phenotype that's been characterized has been very little done on it.
Yeah. I have more questions if that's okay, unless someone else wants to jump in.
Yeah, please go ahead.
Okay, yeah. Just hold on, lost my train of thought. I've always found it fascinating because I mean it's a gene of endogenous retroviral origin. It's unique to vertebrates. Was it ever a problem for our immune system to deal with, and now it's such an important gene. So I'm just really curious how maybe our immune system had to adapt to using what was previously a foreign entity.
Yeah, no, that's really interesting. In fact, I think the evolution of ARC's taken a long time. So it's 400 million years since we think that germline happened, that retrotransposon getting into the germline. But there are some more recent events. A lot of mammals have these gag genes that are more recent retrotransposon sources, and even, of course, you have the so-called endogenous retroviruses, which are actual viral infections that have become dormant in the germline, and that are remnants of infections prior to the species evolving. And it's not clear, whether those endogenous retroviral sequences can get re-expressed, but there's certainly the case that it looks like sometimes in disease context, you can get the re-expression of these things. And so in those cases, they're potentially harmful and there could be an immune reaction. But there is sort of this, interesting then question of whether if you misexpress these these endogenous gag-like genes, do you get some sort of immune reaction or not?
Yeah, definitely.
Thank you very much, Philip. I think I have a question. What's really fascinating for me is like, why do they need both capsid and plasma membrane or extracellular vesicles to transmit the genetics materials? It seems to be like double the work to me, or I don't know. Do you have comment on that?
Yeah, we are still trying to figure that out. So one is sort of easy explanation is that the membrane has proteins that can specify, where uptake occurs. And that's, of course, how HIV specifies that it can get into the T cell with the envelope protein. But you're right in that, viruses have solved the ability of the lipid barrier to get through it, because these envelope proteins are fusogenic. They open a hole. One of the big sort of critiques or mysteries in the extracellular vesicle field is how do you get robust transfer of anything if you don't have a fusogenic protein? And a lot of virologists always ask me, like, well, how does this membrane fusion work? So we don't know yet. I would say that having a membrane can certainly help dictate cargo and specificity of where it gets taken up. But and of course, the other thing that happens for viruses is that the membrane protects the capsid so that the capsid does not induce immune response. And this could go back to Philip's question about tolerance, and maybe you need a vesicle, you need a membrane around the capsid, otherwise the immune system's going to go, oh, they've got an infection.
Yeah. I guess also like with the current updates on the EV field, where people start to look into heterogeneity of the vesicles, it will be quite interesting to find out whether the same vesicles that carry the ARC protein also carry the mRNA, and whether there is a difference between those two. I think there's a lot of things. Well, you already listed like already 10 or maybe 20 questions.
It's just busy.
All right. Is there any other questions? No? Philip?
Wait, I would throw something out that in terms of, this is one indication where there may be a very specific protein RNA interaction moving into a vesicle. We talk a lot about this in the EV field, and for the most part, we don't have any great idea how particular RNAs get loaded.
Yeah.
Or, I mean, what do they bind to? What makes them appropriate, so to speak, to get into a vesicle? In this case, you bought it. It's right there. So I would argue it's a very specific way to get an RNA in.
Agree. Because we obviously sort of thought long and hard about why you need a capsid rather than just have a vesicle. And that is one major advantage, is that the capsid itself already sort of allows specificity in loading of specific cargo that protects that cargo. And especially if maybe, if it's a long distance queue, long distance signal, you want to do that. But I think, yeah, the specificity could be built in already with the cargo.
Is the ARC protein in EV functional? Is it tested yet?
Yeah, so we're doing some of those experiments now, and I would say yes, it does seem that if you concentrate extracellular ARC containing extracellular vesicles, put them on neurons, you actually get this decrease in synaptic receptors that just like the endogenous protein does. And so we think the protein is functional. Right now with that experiment, we're still trying to see if it's because is it due to the capsid disassembling, and so you got a bunch of protein, or is it the RNA has to get translated first and then you get an effect?
Yeah, very interesting. Philip, another question?
Not extracellular vesicle related, so I'm gonna wait until everybody's gotten their EV topics.
Michael, no?
I think
Okay.
I've talked about.
Yeah, okay. I think that's really great, fascinating. Well, with the time also reaching 1 hour, I think I'll conclude this session, and I'll thank very much Jason for sharing your result and also answering all the questions. That's really exciting discussion today. Thanks so much for that.